is part of a lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. The gene construct is a synthetic DNA sequence that will be inserted in the genetically modified organism. It contains at least 4 elements: a promoter sequence, which indicates and regulates DNA transcription, one or several genes of interest coding for traits that the breeder wishes to improve, a reporter gene, which indicates whether the insertion of the gene construct was successful, and the terminator sequence, which indicates that the transcription of the construct should be stopped. Here we will see in detail the roles of a promoter and terminator, which initiate and end DNA transcription respectively. At the very minimum, a promoter contains a transcription start site and the site for binding transcription factors and RNA polymerase. 7 different transcription initiation factors are recruited and bound to the promoter site before the RNA polymerase can be recruited and activated. Once activated, the RNA polymerase will move downstream from the promoter, copying the DNA information into an RNA molecule until it reaches the terminator site. In addition to indicating the start of DNA transcription, promoters can also regulate the expression of the gene. This is done with upstream regulation sites. Based on their activities, we distinguish 3 different types of promoters. Promoters which induce constant DNA transcription in all cells at all times are called constitutive. They are usually associated with housekeeping genes that need to be expressed at all times so that the plant can function normally, such as ribosome production genes. Constitutive promoters are pretty universal across species or even higher taxa, meaning that they can be easily used in transgenesis. However, a permanent expression of a gene can tire out the plant because it requires constant use of its resources. Other promoters can be tissue or stage-specific. Tissue-specific promoters are only expressed in some tissues of organs. The Bt gene, for resistance to bollworm, needs to be only expressed in the fruit, which is the only part of the plant that is attacked by bollworms. Alternatively, a tissue-specific promoter can affect green plant parts, but not the flowers and fruits, especially if they are destined for human consumption and cannot be genetically modified because they underlie strict regulations. An interesting example about a tissue-specific promoter is the promoter PCL that is only activated in the tubers of potatoes. The promoter was used to produce potato crops that are not affected by cold sweetening. Cold sweetening occurs when an enzyme called invertase is activated by low temperatures and it hydrolyzes complex sugar molecules into simple sugars. These simple sugars then easily caramelize when exposed to high temperatures, for instance when potatoes are fried. Genetically modified potatoes that contain a nonsense invertase gene associated with the PCL promoter have a normally functioning invertase in the green parts of the plant, which is key for energy production of the plant, but the invertase is not active in the tubers, which can then be stored in cold for long times without it affecting the quality of the fried potato products. Stage-specific promoters, rather than being activated in a particular tissue or organ of the plant, are activated during a specific period of its development. For instance, they can be associated with genes that accelerate the growth of young plants and are then deactivated when the plant reaches maturity or senescence, so that no genetically modified product is present in the plant at the time of its harvest. Finally, inducible promoters are activated when the plant is exposed to some external physical or chemical treatment. Inducible promoters are particularly interesting for the engineering of plants with better defense mechanism against pathogens, as these defenses are only required when the plant is being attacked by the pathogen and are consuming valuable resources otherwise. An inducible synthetic promoter, called parsley D, was used to engineer canola crops resistant to Sclerotinia sclerotorium, a fungus which can decimate canola harvests. Different promoters were associated with a chitinase defense gene. Plant A, that has not been engineered, is highly sensitive to the fungus. Plants B and C that were engineered to contain a chitinase defense gene are less vulnerable to the fungus. Plant C, which has an inducible promoter, has even higher resistance to the fungus, probably because chitinase production can be adjusted to the presence of the pathogen. So promoters are DNA sequences at the beginning of a gene construct that initiate and regulate DNA transcription. They can be constitutive, inducing DNA expression in all cells at all times, tissue or stage specific, and be active only in certain plant organs or at specific developmental stages, and inducible and be active only after the plant is exposed to a chemical or physical treatment. Terminators are DNA sequences at the end of a gene construct and indicate where the DNA transcription should stop.